Hello everyone. In the previous video we were talking about the mRNA, the transcription, translation, tRNA and ribosomal RNA. Let's talk about the process and what are the molecules for this question 16 here are involved in each process. In transcription, what are the molecules are involved? The DNA is involved in the transcription and the mRNA is involved, both of them. And the information will be copied from the DNA to mRNA. What is the product? The product here, which is what? mRNA, right? So here the product is mRNA. mRNA. In the last the translation, mRNA, tRNA and ribosomes, and the product here, what is the protein, right? It's the translation process of making protein, a process of making a protein. I hope this one helpful to understand. Let's talk about the mRNA, tRNA and RNA, the function of them. The mRNA, it's what? It's the copy information from the DNA, right? And where it happens inside, it will be transcribed where? Inside the nucleus. Then it will be released where? To the cytoplasm. The tRNA and ribosome, it has ribosomal RNA. It attached with the RNA. And the tRNA, what is the function of it? It carries what? It carries amino acid. You remember we said it has the amino acid and has the anticode. Okay, so this is for what? For the tRNA. Okay, so here let's talk about the function of each one of them. I'm going to write them here. Let's start with the first one, the mRNA, the function of it. I'll make it as a table, which is better. Okay, this is a table. What is it and the function of each one. I'll start with the first one, which is mRNA. Okay, what is the function of it? Here provides instructions for building a protein, right? Okay, what about rRNA, ribosomal RNA, rRNA, ribosomal RNA? What is the function of it? Makes up the ribosome. It helps to mix up, because the ribosome, it's made up of protein and ribosomal RNA, makes up the ribosome. Okay, tRNA delivers what? The amino acid, right? tRNA, the function here, transfer RNA, transfers amino acid to the ribosome during translation. Here you have to differentiate between these three RNA and the function of each one of them to make a protein. Okay, let's look at this one here. Listen to this video here. Here, just to remind you about the replication, making copies. You have to know now what is the replication, transcription, translation. These words are very important to know them. Look, listen to this carefully.
Cells in your body are dividing constantly. Every time a cell divides, its DNA must be copied. The process of copying DNA is very fast. Human DNA gets copied at a rate of several thousand bases per minute. The process of copying DNA is called replication. During replication, DNA produces two identical copies of itself. Click the picture showing DNA unzipping to learn more about replication.
So this one, I'm going to show you about the unzipping first, how it will open to make the replication. Then we will talk about transcription and translation, okay?
During the first step of replication, the DNA molecule unzips or splits down the middle where the bases meet. A helper molecule called DNA polymerase attaches to each unzipped strand. DNA polymerase aids in replication.
The helicase, which is unzipping enzyme, DNA, the polymerase that will build, if you remember. Look at this.
The helper molecule slides along each strand, allowing nucleotides with complementary bases to be added. During this process, nucleotides with adenine bases are added to the new strand, opposite to nucleotides with thymine bases on the old strand. Nucleotides with thymine bases are added to the new strand, opposite to nucleotides with adenine bases on the old strand. Nucleotides with guanine bases are added to the new strand, opposite to nucleotides with cytosine bases on the old strand. Nucleotides with cytosine bases are added to the new strand, opposite to nucleotides with guanine bases on the old strand. After all complementary nucleotides have been added, two identical DNA molecules are formed. The helper molecule detaches. Each new molecule contains one strand from the original DNA molecule and one new strand. Each molecule can split again when it needs to be replicated.
Okay, so this one here for the copying DNA or replication of DNA before the cell division. Listen to this. You remember the mutation when there is a problem?
DNA doesn't always get copied accurately. Random errors occur, or DNA that is damaged by UV light, chemicals, or other agents can experience copying errors. Cells produce proteins that can fix some errors in DNA. However, sometimes a mutation occurs that is not fixable. A mutation is any change in the bases of a DNA molecule. Some mutations have no effect on a cell, and others can be beneficial. But some mutations can cause diseases. A genetic disorder results from mutations that affect the normal function of a cell. Some of these disorders, such as Tay-Sachs disease and sickle cell anemia, are inherited or passed on from parent to offspring. Other genetic disorders result from mutations that occur during a person's lifetime. Most cancers fall into this category. Click the pictures to learn about the different types of mutations.
Okay, let's start with the first mutation, first type.
A deletion is a mutation in which at least one base pair is removed. A deletion is like removing letters from the middle of a sentence. A deletion, like any other type of mutation, can cause a gene to code for the wrong protein. An insertion is a mutation in which at least one base pair is added. and insertion is like adding extra letters into the middle of a sentence. A substitution is a mutation in which a base pair is replaced with a different base pair. A substitution is like changing a letter in the middle of a sentence. Substitutions are the most common type of mutation. Sickle cell anemia is caused by a substitution mutation.
Okay, let's continue. These are about the mutation, just review.
Proteins determine a variety of traits and act as triggers for many cell processes. DNA provides the instructions for making proteins. But since DNA cannot leave the nucleus, it needs other molecules to actually build proteins. RNA, or ribonucleic acid, helps with the process of protein building. RNA is very similar to DNA with a few subtle changes. One change is that RNA contains the base uracil, U, instead of thymine, T. In RNA, adenine, A, pairs with uracil, U. The first part of the protein building process is called transcription. During transcription, a single-stranded molecule called messenger RNA, or mRNA, copies instructions from DNA and moves these instructions out of the nucleus and into the cytoplasm. After transcription, an mRNA molecule leaves the nucleus and moves into the cytoplasm of the cell. The process of translation can then begin. Click the picture to see how the processes of transcription and translation can produce a protein.
Look at this one here, how the ribosome are attached with the mRNA.
During translation, the mRNA is fed through a ribosome, which is a cell organelle composed of protein and ribosomal RNA, or rRNA.
Look at this one here. This shows the transfer RNA. This is the transfer RNA, and these here represent the amino acid they are carrying, and this is the anticode they have here down, how they are matched with the other side of the complementary mRNA. Okay, look.
The mRNA moves through the ribosome 3 bases at a time. Each group of 3 bases codes for a specific amino acid. Molecules of transfer RNA, or tRNA, that have complementary bases match up with the mRNA and deliver amino acids to the ribosome.
Look at the end.
The ribosome then joins the amino acids together to make a protein.
Okay, so this is all about the transcription, translation, replication, mutation. If you still have any question about these, you can send me your questions. Thank you for listening.